Okay, let's start with the interaction of follicle cells in the germline. So we will start again in the germarium, and we know that between region 2A and 2B the follicle stem cells are located, which give rise to different forms or different types of follicle cells that are necessary to encapsulate the germline cyst, leading to a disc-shaped germline cyst with the oocyte in the center, and that's where we'll start off. Now, this disc-shaped germline cyst with the oocyte in the center will now experience a force and a few different things. And that is the stalk that will be formed between this germline cyst and then egg chamber and the next posterior, more advanced one. So let's just quickly remind us that we have here, let's say in region 3, there is an egg chamber, and there is a stalk to the next egg chamber in stage 2. And that is because this egg chamber in stage 2 made a stalk to next newer anterior egg chamber. And this stalk does two things. Firstly, the germline cyst, the more anterior germline cyst, the newer germline cyst, will round up and be sphere-shaped. The second thing is that the oocyte is not located centrally anymore, but posteriorly. That is the first asymmetry within the germline cyst, so to speak. And that occurs just simply by the formation of the stalk. So, very important, the posterior, more advanced egg chamber always signals to the newer, more anterior egg chamber and leads to the posterior localization of the oocyte and also to the sphere-shaped germline cyst rather than disc-shaped. And we'll look now exactly at this interaction, whereas we start at what happens anteriorly of a germline cyst at region 2B and 3, so when encapsulation takes place, and what the first signaling pathways are, and we concentrate on the anterior side because that's where the stalk will be produced by this egg chamber. So let's just imagine this is the germline cyst in black. Then you have the follicle cells, different kinds of follicle cells here in these squares here. And they will then here produce the stalk to the next newer germline cyst, because this one, will move more posteriorly and also the others. So let's start within region 2B. Encapsulation takes place and we have different kinds of follicle cells. We have the polar stalk precursors here in green, and we also have the epithelial follicle cells then also joining this encapsulation event here in black. The polar stalk precursors, as we already said, are also sometimes named after their migrating behavior. Then you would call them CMCS or PMCS. CMCS for cross-migrating cells and PMCS for posterior migrating cells. In this case, these green ones are the cross-migrating cells, because here you would say, well, out here the follicle stem cells are located, and they will then the follicle cells that are coming from these follicle stem cells will migrate down here. That's why it's called cross-migrating. And also you would have here some follicle stem cells where others migrate back to the germline cyst, which is not drawn here. And then they are called posterior migrating cells. But that's not so important. Important is that these are the polar stalk precursors that are different than the epithelial follicle cells, which are other follicle cells. Now, the first signaling will be delta notch signaling. So it's all about delta notch here. The germline cyst expresses normal levels of the delta ligand. Now, the notch receptors here on these cells will receive the delta ligands, so the delta signal. So every cell here receives the equal levels of delta. However, there's a difference. The polar stalk precursors express fringe, whereas the epithelial follicle cells do not express fringe. And that's the main difference. Whenever a cell expresses fringe, the notch receptor is more sensitive towards delta. That means that these two green cells here in contact with the germline cyst receive more delta notch signal than these black cells here. So you could say these black cells receive medium amounts of delta notch signaling, these as well, whereas these two green cells here receive high levels of delta notch signaling, because they express fringe and they're more sensitive. So what about these four cells here? these 4 green cells, they are not in contact with the germline cyst. They are, of course, not only 4, I think there are 6 to 8, but these are also polar Stalk precursors, and they receive different signals. They also receive delta notch signaling, but not by the germline cyst, but by other follicle cells. So let's say these cells here also express delta, and in that manner signal to these next ones here, and they also express delta signaling to the next, and so on. So they also receive delta notch signaling. However, follicle cells express the metalloprotease kuzbanian-like. That means that they only express low delta because delta is cleaved or inactivated. And in that manner, they only receive, so these 4 here, only receive low delta notch signaling. So this is very important. You have here medium in these 2 black ones. You have also medium levels in these 2, so the epithelial follicle cells. You have high levels in these 2 cells, so in the polar Stalk precursors connecting the germline, and in the polar Stalk precursors not connecting the germline, you have low levels of delta notch signaling. So 3 different amounts of delta notch signaling. Now, the first differentiation event occurs through that. The cells, the polar Stalk precursors with high delta notch signaling here, will in fact differentiate into polar cells. So these 2, which are in fact 3 to 5, will be anterior polar cells and will differentiate as anterior polar cells. And as we'll see, that is because high delta notch signaling is usually connected to a differentiation event. These ones receive medium delta notch signaling, which is probably not enough to make them differentiate. That's why they stay immature. And in fact, epithelial follicle cells will stay immature until stage 6 to 7. So they will not differentiate. So they are also not important yet. We just talk about the polar Stalk precursors now. So polar cells, anterior polar cells differentiate. So let's look at this picture here. Here we have these 2 blue cells, which are now the anterior polar cells. They have differentiated. This is actually what I mean by this sign here, this red one. This is always when cells differentiate. Okay, so we have these blue anterior polar cells which have differentiated, and what they do now is they will express or secrete a factor which is called unpaired. That's the second signaling pathway, JAK-STAT. So the JAK-STAT pathway is used here. They secrete unpaired in all directions, and every cell that contains the Domeless receptor will receive the signal. In fact, all follicle cells are proposed to have this receptor, Domeless, so all should have now unpaired signaling or JAK-STAT signaling. However, there's a difference. Although each cell receives the signal, they will not be affected by the signal. So not each cell will be affected by the signal. And the reason why that is because you have here, let's say delta notch, and then you have JAK-STAT. And delta notch in fact inhibits JAK-STAT, and JAK-STAT inhibits delta notch signaling. So if you have cells with high delta notch signaling, JAK-STAT signaling is not possible. And vice versa, if you have high JAK-STAT signaling, delta notch signaling will not be possible or will be inhibited or interfered with. So because there are high levels or medium levels of delta notch in these cells connecting a germline cyst, there will not be JAK-STAT signaling. And that is in fact because the STAT protein, the signal transducer and activator of transcription that we talked about, will not be nuclearized. And because this relation here, this inhibitory relation, is not universal, it is proposed that probably there is a soluble protein that maintains this interaction, because later delta notch and JAK-STAT will not inhibit each other anymore. But that's later. So now they're inhibiting each other. So only cells receiving low delta notch are able to receive the JAK-STAT signaling. And that's these cells here. These, in this case, 4 cells, the Stalk or polar Stalk precursors not receiving high or medium but low delta notch are able to respond to JAK-STAT signaling. And that leads to these 4 cells now differentiating as Stalk cells. So low delta notch plus JAK-STAT signaling leads to Stalk cell fate if there are polar Stalk precursors. Okay, we talked about what happens anteriorly of such an egg chamber. Now let's switch it up and talk about what happens posteriorly of such an egg chamber. And for that we need to discuss the interaction between 2 egg chambers next to each other. So let's call this egg chamber here NA for new and anterior, and here this one we call OP for old and posterior. So this one is the new one and this is the older one, or so that would be timely, or we can look at it spatially, and this would be the more anterior located one and this would be more posterior, again depending on the location of the ovariole. Now in blue again, the anterior polar cells here, usually 3 to 5 at this stage, but later on there are only 2 because the other one will undergo apoptosis. You will have the Stalk cells here in green, and yeah, so we talk about the interaction, and we already talked about that this older egg chamber always influences the newer egg chamber. This newer one would then influence the next newer one, and so on. So they are located initially here in sorry, not stage, but region, here region 2b, and here would have region 3. And then, while this stalk formation happens, while these stalk cells or these stalk polar precursors change from immature to differentiate into actual stalk cells, this NA goes from region 2b to region 3, and the OP goes from region 3 and hatches into the ovariole into stage 2. So hatches into the vitellarium from the germarium. So that is a movement that occurs while this stalk is formed, and then you have exactly this picture. You have one egg chamber here in region 3, the stalk, and then the next egg chamber, the OP, in stage 2, and here it already occurred in these ones. So this is the prime interaction here that is really important. So what occurs is the stalk formation. The stalk formation leads to 2 things. Firstly, a force, an integration force, and then that leads to a sphere-shaped germline cyst here. Here it already happened, because this one was already influenced by the older one. So sphere-shaped, and the second thing is that these 2 cells here, which again are about 3 to 5, but later on 2, are actually polar cells, posterior polar cells, and to be exact, they are prospective or future posterior polar cells, because right now they're also here drawn in green because they are proposed to differentiate first as stalk cells, here also in green. That means they upregulate E-cadherin adhesion protein in these 2, and that's why these 2 cells here in this pool have higher levels of E-cadherin than these black cells, so the epithelial follicle cell precursors. And for the oocyte there is the same. The oocyte produces more E-cadherin or expresses more E-cadherin than all the other cells of the germline, all the other nerve cells. And then something occurs that is called preferential adhesion, and that means that the cells with most E-cadherin will bind to each other because that's what is energetically favorable, to exchange lower binding states to more higher binding states, more force. So that is what occurs here, preferential adhesion between the oocyte and these stalk cells, which are in fact prospective posterior polar cells. And that leads to the posterior localization of the oocyte. That's the second thing that occurs through stalk formation. And then here in red we still have such a structure that we already called the Balbiani-body. These things, or this red color, should indicate that this is an accumulation of different factors like RNA, proteins, and organelles, and that is because there the MTOC is located, the minus ends of the microtubules is located, or are located, and the microtubules extend in these nerve cells, and things are transported to this area here. And what happens now in late region 3 is that this red structure will be located more posteriorly. So I will draw that here. Will be located more posteriorly within the oocyte. It is not exactly clear why that happens, or I should say how that happens, but what is known is that a protein called PAR1 is necessary. So whenever you mutate PAR1, this so-called posterior enrichment does not occur. And it is a very likely candidate to be responsible for that, because PAR1 was initially found in C. elegans, and there it's also located posterior and is in fact necessary for the AP axis in C. elegans. And here the same seems to be the case. There are two things quite known about PAR1, and that is that it is proposed that it might be also located here posteriorly through a so-called stimulus, a mechanical stimulus of the follicle cells. That could be part of this puzzle, what PAR1 exactly does or how it's activated in late region 3, how it is here activated in the posterior region and leads to the posterior enrichment. So mechanical stimulus maybe by follicle cells, or the other theory is that it is firstly, what they saw is it's firstly found within the fusome or located by the fusome. It is then also found here at this Balbiani-body anteriorly and later relocates to the posterior area. So that is also something that is known about the PAR1. However, whatever the mechanism is, it is responsible for posterior enrichment, and when you mutate it, it doesn't occur. So let's paint a coherent picture one more time. Imagine having one egg chamber. We discussed what happens anteriorly and we discussed what happens posteriorly. When having one egg chamber, the anterior stuff happens throughout region 2b and 3. So because the egg chamber always moves through the stages and through the regions, of course. What happens posteriorly is mainly important starting with region 3 to stage 2 then. So anteriorly in region 2b and 3, what happens is basically a preparation for stalk differentiation. So all these things occur, but the stalk hasn't differentiated yet. But everything is basically prepared for that. At the posterior end, we start with region 3 at the same time, basically, while that all happens. And in region 3, the stalk by the next older one is produced and influences now our egg chamber and shapes these posterior ends here, or it's this posterior end here, and leads to sphere-shaped Germline cyst and to the posterior localization of the oocyte. At the same time, also the Balbiani-body is transferred to posterior enrichment. Now, at the anterior end, like I said, the whole thing is prepared, and then our egg chamber hatches from the germarium into stage 2, while now differentiating the stalk to the next newer one. And the next newer one already also prepared for the stalk differentiation and is now influenced by this stalk again. And now these posterior enrichment also occurs, and the posterior localization of the oocyte and the sphere-shaped germline cyst. And now this one hatches again and produces the stalk and influences the next newer one, and so on. Now we're finished with what occurs within the germarium, and we basically explained how the first asymmetry within the germline cyst arises, how the oocyte is located posteriorly, how the inner polarization takes place, and this information now, this posterior localization, which is basically an information about where is posterior and where is anterior, will now be located to the follicle cells and then located back or transferred back to the oocyte, creating the anterior posterior axis as well as the dorsoventral axis. And that is now what we discuss throughout the next stages. Alright, so now we're going to discuss what happens within the vitellarium at early stages, early oogenesis. So now we can use these terms again, early oogenesis within the vitellarium. That's where we are right now. So between stage 2 and stage 6 to 7, we'll see. So three things occur. Firstly, the polar cells, the prospective posterior polar cells, will differentiate into polar cells. Here there are still stalk cells within the germarium, but they will differentiate now. Secondly, polar cells will undergo apoptosis, So they are not 3 to 5 cells anymore, but 2 cells. Only 2 cells will survive. Thirdly, the epithelial follicle cells here in black will differentiate into terminal and central follicle cells. So let's have a look at that. Firstly, the prospective posterior polar cells will differentiate into posterior polar cells instead of Stalk cells. So they will change their fate. And that occurs, or is proposed to occur, by a spatial event. So at the beginning, they seem to not really be too much in contact with the germline, receiving not so much delta notch, but receiving a lot of JAK-STAT signal from here, from these anterior polar cells of this egg chamber here. And because they still inhibit each other, delta notch signaling not really can occur here, but JAK-STAT signaling, and that makes them Stalk cells. However, when this whole spatial thing changes throughout these stages, they will contact, so these two cells will contact the germline cyst more, and thus the delta notch signal will be more prevalent than the JAK-STAT signal, leading to then JAK-STAT being inhibited and delta notch being activated. So then they, here at the beginning of the vitellarium, they're going to differentiate into posterior polar cells. Now they behave just like the anterior polar cells and secrete unpaired in every direction. However, the epithelial follicle cells cannot respond to it because they are still medium, at least, in delta notch signaling, and that inhibits each other. So yeah, also these cells are only 2 anymore, so the polar cells diminish from 3 to 5 on each side now to 2 on each side. Okay, and now the epithelial follicle cells. They cannot receive these unpaired signals because they have medium delta notch. But now 2 things occur. Firstly, JAK-STAT signaling and delta notch signaling does not inhibit each other anymore. Suddenly, that's not the case here. So both can occur at the same time, and that's what is proposed, or that's why it is proposed, that there is probably a soluble protein or molecule that is responsible for this interaction that is now not expressed anymore by these follicle cells, and now they can receive both. Alright, so now they can receive delta notch signaling, but medium delta notch signaling is not enough to differentiate. They need high delta notch signaling just as the polar cells, but they have only medium because they do not express fringe, so their notch receptor is not that sensitive. So what happens at stage 5, beginning at stage 5 and peaking at stage 7, the delta ligand within the germline cyst is upregulated, is higher. And now these epithelial follicle cells receive high delta notch signaling even though they do not express fringe. So now they're ready to differentiate. And it depends in what they differentiate, depends on which signal they receive. And now they're able, because this interaction is not happening anymore, they're able to respond to this unpaired ligand on each side here in blue. So, generally speaking, these epithelial follicle cells now stop to proliferate. They usually proliferate starting with the germarium until stage 6 to 7, 5 times mitotically. Now they stop and begin to endocycle, so they replicate the DNA without proliferating actually, so they get polyploid just as the nurse cells. So mitotic to endocycle switch, that is what occurs around stage 6 to 7, mitotic to endocycle switch of the epithelial follicle cells. Now that they're ready to differentiate because they stop proliferation, that's usually inhibiting each other, so either proliferation or differentiation. Once proliferation stops, differentiation occurs. That's a general principle usually. Alright, so differentiation can now occur, and that's dependent on which signals they receive. So now let's see. These signals receiving the unpaired soluble protein are able to nuclearize the STAT protein, and in that manner they will be terminal follicle cells. So all the cells receiving JAK-STAT will be terminal follicle cells. Cells receiving no JAK-STAT will be just central follicle cells. So here central and here terminal. And now a third signal next to delta notch and JAK-STAT plays a role, and that is Gurken, or TGF-alpha. TGF-alpha is located here at the posterior end of the oocyte. We know through posterior enrichment that we already know in region 3. And this Gurken signal, or TGF-alpha signal, is only here at the posterior end. So these terminal cells also have this signal leading to posterior terminal fate. So the default fate, the default terminal fate is anterior, here. And the fate of terminal cells, if Gurken or TGF-alpha is received, is posterior terminal fate. So that is what's meant by this transfer of information. So the oocyte transfers basically the information about being posterior to these terminal follicle cells. Now they know they are posterior. They basically know that they are posterior terminal follicle cells, and these are anterior terminal follicle cells. And that is what I have drawn here again. So here we see what they differentiate into. Black is the central follicle cells. Then we have here the anterior follicle cells in 3 colors because they differentiate into 3 different types of cells. And that is also an interesting thing, that here the anterior polar cells act as a morphogen. So they have a diffusible factor and it's concentration dependent. So unpaired is concentration dependent on which cells they are going to be. So high levels, medium levels, lower levels. 3 types. And at the posterior terminal cells, at the posterior end, this is their fate here in violet, but they do not differentiate into different cells. They're just one cell type here. Okay, let's have a look at delta notch signaling one more time. Usually, it is the case that delta notch signaling, when the concentration is high, when delta notch signaling is high, it leads to differentiation and stop of proliferation. That is what causes the mitotic-to-endocycle switch. That is what causes this differentiation of the anterior polar cells and also of the posterior polar cells. However, in this case it might be true, in others it is not. So it depends on the history of the cell, on the gene regulatory state of the cell, how it responds to the delta notch signaling. So in this case, when we, for example, mutate notch of the notch receptor of these epithelial follicle cells, they are not able to differentiate and they will keep dividing through these stages. So that indicates already that double notch signaling is necessary for differentiation. However, in the stalk, it plays a different role. We saw that when the stalk differentiates, it needs not only JAK-STAT signaling, but also delta notch signaling, but only in very low levels. So how is that possible if high levels lead to proliferation? Because in the case of the stalk, delta notch signaling is not responsible directly for differentiation, but has probably a different function. And the function in this case is proposed to be that delta notch signaling keeps cells viable. It's kind of a signal that prevents apoptosis of polar stalk precursors that will be stalk cells. And that is because that limits the amount of cells that can form a stalk, and that is producing always the same amount of stalk cells. So that notch signaling is here responsible for determining how many stalk cells can be kept viable with a specific amount of delta that is expressed. Because the more cells you have, the less delta can be bound to each cell, and that keeps these cells then not from being viable anymore. So they will die until there is enough delta concentration per cell to let them survive. So that is what is proposed here to be the function of delta notch signaling. To illustrate the connection between delta notch and the stalk cells, let's have a look at some mutant phenotypes. Whenever you upregulate just slightly notch in prospective stalk cells or delta in anterior polar cells, you will get a longer stalk because more cells can be kept viable because it's not that low. Delta notch signaling is then a little bit higher. However, if you overexpress delta to a greater extent in anterior polar cells, you will receive higher delta notch signaling. And if it's too much, then JAK-STAT signaling is inhibited and no cells can be stalk cells. They will all get polar cells, in fact. So you get more polar cells and less stalk cells. However, whenever you look at anterior polar cells and mutate delta, so it's not expressed at all, there is no delta notch signaling with these stalk precursors, and then no stalk is produced at all. So to summarize the interaction between germline and soma, between the germline cyst and the follicle cells, we start within the germarium, where encapsulation takes place. And in fact, encapsulation is also partly led by delta notch signaling. However, we haven't really mentioned that. Delta notch signaling can be divided into early delta notch signaling and late delta notch signaling. We discussed early delta notch signaling, which is responsible for differentiation of the anterior polar cells. Because anterior polar cells come from polar stalk precursor, which have or express fringe and therefore can have high levels of delta notch when in contact with a germline cyst. Polar stalk precursors not in contact with the germline do not receive high delta notch signaling, but only low delta notch signaling because they are in contact with anterior polar cells or other polar stalk precursor, which altogether express kuzbanian and thus only low delta. That is early delta notch signaling and responsible for anterior polar cell differentiation and also for stalk differentiation. The other thing is late delta notch signaling. Late delta notch signaling is responsible for differentiation of the epithelial follicle cells later around stage 6 to 7 within the vitellarium and not within the germarium. Within the vitellarium, around stage 6 to 7, we have talked about the epithelial follicle cells that differentiate into terminal and central follicle cells. And at the end, the terminal follicle cells are again split up into anterior and posterior, whereas the anterior is the default fate, whereas posterior is what is occurring only when the oocyte has migrated posterior, as well as the Balbiani-body has relocated posteriorly. And this positional information is then transferred onto these posterior located terminal cells. Then in between these two, between early and late delta notch signaling, the posterior polar cells are also, well, differentiating before they differentiate into or differentiate as stalk cells, and then around these stage 2 and 5 they differentiate into posterior polar cells just like the anterior polar cells. We discussed which signals are responsible for all these different types, and we also discussed that, or what we see now is that the anterior polar cells also work as kind of a signaling center, whereas the posterior polar cells do not. Why signaling center? Because they are affecting the fate of different cells around them in a morphogen manner. So in a concentration dependent manner, these cells around them differentiate into different cells, as well as the stalk here around here. Alright, now we can move to the last part, and that is the follicle cells that retransfer the positional information to the oocyte. The oocyte is then able to restructure its cytoskeleton and is then able to build the anterior posterior axis and the dorsal-ventral axis. So the posterior follicle cells are now signaling back to the oocyte and restructuring the cytoskeleton. In fact, they're doing it the following way. We have minus ends here anteriorly located and plus ends posterior located. Motor proteins like dynein or kinesin can now travel in specific directions. So dynein, for example, from plus to minus, and kinesin the other way around. Whenever molecules associate with these motor proteins, they will travel to their specific poles. And this asymmetry in molecule distribution is the beginning of the anterior-posterior axis and the beginning of segmentation, which is then of course way more complicated. But this is the very beginning, which will be important for early embryonic development. At the same time, next to this thing here, to the cytoskeletal rearrangement, we have also the other thing, which is the nucleus moving from here, from posteriorly to the upper anterior margin. At the same time, you have this Gurkin TGF alpha homolog located around the nucleus now, so from posterior also to the upper anterior margin. And it's probably located there because of some microtubule system, because that is not the only thing. So it's not only structured like this, but there is a more complicated behavior of microtubules here, because the nucleus also serves as an MTOC, as a microtubule organizing center. And in that manner, Gurken is somehow transferred to this area here. And again, just as we had here with the posterior follicle cells, we also have the same event happening here with Gurken or TGF-alpha. It is again secreted to the space here between the follicle cells and the germline cyst and makes these follicle cells dorsal. The follicle cells around here will be ventral by default, and they will again signal back the positional information back to the oocyte in embryonic development later. So the same event happens here. So here we had the oocyte transferring information to the follicle cells and then back again, and we have the same here again. The oocyte transfers positional information to the follicle cells, and then the follicle cells later embryonic development will give that information back to the oocyte, and in that manner structuring the dorsoventral axis with the mesoderm and the ectoderm and the amnion serosa and so on. So the only question that is still here is what I tried to indicate with this question mark: if these both events are if there's only a correlation or if there is a causation. And in fact, this is not known. So it could be that both come from a single signal or from two signals. However, both seem to be dependent on this event here. So if the posterior follicle cells are not determined, so if, for example, the oocyte is not located posteriorly, and thus the posterior follicle cells are not defined because they do not receive the Gurken or TGF-alpha signal, then this restructuring event takes not place, and the Gurken and nucleus relocalization here does also not take place. So both seem to be dependent on this asymmetry here, so that the posterior follicle cells become or receive the positional information from the posterior located oocyte. So to come to an end, last things that occur are, as we already talked about, vitellogenesis. So follicle cells around the germline cyst produce yolk proteins, and in fact they even have a lot of ER at this stage. So that is something that we learn from these follicle cells, that ER can be, so the endoplasmatic reticulum, can be upregulated, can be produced in higher amounts when necessary. And in this case, they need to produce a lot of yolk compounds, a lot of yolk molecules, and in this case, they then also have high amounts of ER. So the yolk is then exocytosed and taken up by the germline cyst by collagen-mediated endocytosis in form of vesicles. And at the same time, as we talked about, the vitelline membrane is being made by vesicles, which contains compounds of the vitelline membrane. And also, like I mentioned, this is active research still, how these two things can happen at the same time without affecting the other. Then stage 10, we have nurse cell dumping, which is the phase in which then the fast transport arises. So the nurse cells now are transporting all their cytoplasm, or big amounts of it, to the oocyte, and in about 30 minutes, and they also kind of contract. So they have a myosin-actin-based contraction mechanism to really push out the whole cytoplasm to the oocyte, which in this case then also grows significantly. The nurse cells then also come to an end, their life cycle comes to an end, they die by apoptosis, and the oocyte is the last thing that stays. We explained how the AP axis and dorsoventral axis is determined. The chorion is now being made as well. Now we have two membranes, and then it basically hatches from the ovariole, and we leave the ovariole, and it hatches into the oviduct. This is called ovulation, and it is now ready to be fertilized by the spermatheca, a special organ, and is then fertilized by such an opening that is called micropyle, And yeah, then it's fertilized, laid, and then we are already discussing embryonic development, and oogenesis is basically finished.